This real-time sequence shows movement in the heterocont euglenoid Eutreptia. The two flagella are quite different in position, behavior, and function. This sequence shows the volvox colonies actively swimming. They move through synchronized flagella movement that generates strong currents and is able to propel the colony. This is one of the many bizarre organisms that are found in the gut of termites. It is a very large, single-celled ciliate called Deltotrichonympha and is covered with thousands of synchronized flagella. This sequence, speeded up 25 times, shows the motility of the green alga Microsterias, in which mucilage is secreted from special pores. An amoeba moves across the field at 25 times real speed. This cell has eaten numerous green algae, now contained in digestive vacuoles. Movement is generated from cytoplasmic streaming by actin microfilaments. This real-time sequence shows a large euglena, whose cell wall consists of interlocked flexible strips that slide past one another as the cell changes shape. The strips are covered with lumps of polysaccharide. Paramecium bursaria in nature are almost always full of symbionts. These cells are also actively feeding. Many cells can be seen being swallowed, capturing and ingesting other algae. The green algal symbionts supply photosynthetic products to the host organism. This real-time sequence shows reproduction in the green alga Chlamydomonas. Two individual cells are held in contact by their flagella, used as sensory receptors for sexual attractants and determination of the correct mating partner. Cell movement eventually brings a membrane appendage, the fertilization tube, visible between the flagella of one gamete, in contact with a special region of cell membrane in the other gamete, at which moment the cells join. A few minutes later, they are partially merged. In this sequence, speeded up 2 times, a small sperm of the green alga Oedogonium seeks out and fertilizes a large oogonium filled with food reserves. The small active sperm cell is strongly attracted to the big egg cell, whose wall has split slightly to open a fertilization pore. The sperm loses its flagella as it enters the pore and is soon merged completely with the egg cytoplasm. In this sequence, 2 individual Spirogyra algae reproduce via conjugation over about 45 minutes. First, they align and form wall tubes toward one another. Once the connection is complete, one cell, typically the male cell, moves into the other. The resulting diploid cell, a zygote, secretes a thick cell wall. Conjugation is a sexual process of genetic recombination, but is not a reproductive process. That is, no new cells are formed. Red algae are generally assumed to have no motile stages in their complex life histories. However, recent research shows that a variety of different spore types from various red algae have mechanisms for motility. Most of the organisms shown are euglenoids, collected from a stagnant pond. This dinoflagellate, Certium tenu, has two flagella, one very long flagellum, which is not very active, and a spiral flagellum that is tightly wrapped around the cell and that generates most of the movement. The cilia of this paramecium generate strong currents carrying prey to the gullet where they are tasted. The arrows show chlamydomonad cells being ingested into a feeding vacuole, which is pinched off at the base of the gullet and moved into the cell. This is a colonial species of diatom that displays a highly specialized gliding motility, shown here speeded up 5 times. The cells are linking by a tongue-in-groove system along their silica wall. Movement is generated by an actin system coordinated in the colony. While these are protists, they form some of the largest organisms on the planet, specifically the giant kelps of the marine kelp forest. This sequence, showing the release of spores by the red alga Odwinella pacifica, is speeded up 5 times. This sequence, showing the release of spores by the red alga Spiridia, is speeded up 5 times. This sequence of cytokinesis in the green alga Microsterias takes place over the course of about 4 hours. This process involves membrane and cell wall formation. This choanoflagellate, Salpingica species, captures and swallows food. This sequence is shown at real time. These more highly magnified sequences are shown speeded up 25 to 50 times. Arrows indicate the captured food items that are about to be ingested. This acellular slime mold, physarum, is a single cell enclosed by the cell membrane. It grows outwards by rhythmic pulsations of its cytoplasm. These pulses are driven by shuttle, that is, back and forth streaming in the cytoplasm, driven by the actin cytoskeleton. This sequence takes place over 2.25 hours. The cellular slime mold, Dictyostelium discoideum, has 4 different life stages. At this vegetative life stage, unicellular haploid mixamoebas move independently from one another, engulf food particles by endocytosis, and reproduce sexually and asexually. When conditions become unfavorable, mixamoebas aggregate and form pseudoplasmodium or slug. The mixamoebas retain their cell plasma membranes. The aggregation in this sequence took place over about 2 hours. Slug formation took an additional hour. Sequences of the straggler amoebae are shown speeded up about 25 times. Once aggregated, the pseudoplasmodium or slug migrates to a suitable location in order to fruit. This was filmed over about 1.25 hours. The slime mold produces long stalks with fruiting structures at the tip. The cells at the tip transform into thick-walled spores, which are released, and upon germination, release mixamoebas. This was filmed over about 1.5 hours.